So today we are going to discuss in this is our second lecture for the cell suspension culture, where we are going to discuss about the synchronization of suspension culture. So in previous lecture we have discussed about the what is cell suspension culture, by which techniques or what are the different explants or the calyces from which we are able to get this cell suspension culture, and what are the criteria which we have to maintain while proceeding for the cell suspension culture. As we have discussed it earlier, that if we are having friable callus like loosely woven cells, meristematic cells, from that part also we are able to get the cell suspension culture very easily, because when we are placing our friable callus in the liquid medium, due to the shaking, which is like a shaker, we are keeping our explant in the medium onto the shaker at 50 to 150 rpm, which allows cells to loose or get free from the clump. Then these cells are able to grow in the liquid medium. So the like agitation should be such that these cells, the plant cells, should not get harmed up. So due to the shaking, all the cells are continuously in the contact with the liquid medium, so from all the surfaces, so the cells which are in the suspension culture are able to grow uniformly. So this was about the friable callus. If we'll talk about the non-friable callus, it means the cells which are present in the callus are not that loosely woven, or we can say they look like a tough like structure, थोड़ा dense mass होता है cells का. But due to the culturing and subculturing of the callus onto the liquid medium, this non-friable callus is converted into the friable callus. So this is how, again, matlab like there are many angiosperms or gymnosperms from which we are able to get this non-friable callus. But these non-friable callus are not having importance for cell suspension culture. But to extract the secondary metabolites from these non-friable callus, we have to proceed for subculturing of these non-friable callus, so that this non-friable callus is converted into the friable callus. This was the second. And third, the most important, in some cases we are having non-friable callus, but they have also developed the cell wall, as the actively growing cell are not able to form the cell wall. But in certain cases we have scientists observe, like in case of woody plants, the cell, the meristematic cell. Walaikum salam. So again he break my rhythm. So in case of woody plants, what we are having cells with the thin cell wall. So to obtain this, to obtain the cell surface culture from these calluses, what we will do, we treat these calluses with the cell wall degrading enzymes. We will treat with these with the cell wall degrading enzymes. Like we all know that we are having different types of enzymes: cellulase, hemicellulase, pectinases, and we are having many other enzymes with different combinations which can be applicable for this non-friable callus. So even when we are getting the callus, we have to take care the health of this callus in the liquid medium, whether by observing the phenotypic appearance of these calluses, like if there are sometimes these calluses are green, if there is any browning or any color change of the liquid medium, that time we are able to change the liquid medium, so that the callus which or the cells which are in the suspension culture should not get damaged by releasing certain phenolic compounds by the cells. Like again I will say, when the cells are in suspension culture, sometimes there may be some, by any mishap, these cells may get damaged up. So the cells which are damaged in the cell suspension culture are able to produce certain phenolic compounds into the medium, which will lead to the death of other cells, or they may contaminate, or they may degrade our secondary metabolites which are present in the liquid medium of cell suspension culture. So what we do, we can remove these cells easily. First, second is that we can change the medium so that our product will be saved from the degradation. So this is how we proceed for the cell suspension culture. So today we are to obtain or to take the importance of the cell suspension culture. All the cells should be synchronized. Means all the cells which are used for the cell suspension culture should be having or should be at a particular stage of cycle. These cells may be having different size and shapes or different nuclear or the same, they will be having same nuclear content, but when we take the cells, they are when we take the cells from the callus, these cells will be present at the different stage of cell cycle. Like there will be some cells which will be present in the G1 phase, or some will be the S phase, or some cells will be in the G2 phase, or some cells in the medium will be in the M phase. But it means all the cells are working individually. So to obtain a synchronous culture, scientists have given different techniques where we are able to synchronize all the cells all together, and then the all cells will work as a one unit system. मानियो अच्छे वारिया cells तिम क्या अलग अलग different stage में means कभी कभी एक सेल सेकेंडरी मेटाबोलाइट दे रही है, उसके बाद दूसरी सेल आ रही है, वो 2 घंटे बाद सेकेंडरी मेटाबोलाइट दे रही है, तो यह हमारे लिए यूज़फुल नहीं है. तो हम लोग क्या करते हैं, डिफरेंट टेक्निक्स अप्लाई करते हैं इस सेल सस्पेंशन कल्चर को, ताकि जो हमारी सारी सेल्स है जो इंडिविजुअल वर्कआउट कर रही है वो एक यूनिट सिस्टम बनाए और वो हमें प्रॉफिटेबल मतलब प्रॉफिट हमें दे. लाइक we will be able to go for the production of secondary metabolites or even antibiotics. Like first technique where we are able to get the synchronization in case of cell suspension culture is the starvation technique. क्या हम लोग करते हैं, इसमें हम लोग सेल्स को स्टार्व करते हैं और वो जो सारी सेल्स है दे रिमेन एट अ सर्टन सेल डिवीजन. लाइक अगर हम लोग क्या करते हैं, हम लोग इसमें ग्रोथ रेगुलेटर्स नहीं डालते, हम लोगों ने सेल्स ली उसको लिक्विड मीडिया में डाल दिया, वो क्या कर विदाउट द एडिशन ऑफ द ग्रोथ रेगुलेटर्स. what happens, all the cells which are present in the suspension culture will get arrested at either G1 or G2 phase. मीन्स जब हमें लगा कि अब हाँ बाबा सेल ग्रोन नहीं कर पा रही है मीडिया में, what we do then, next step is the we will add certain growth regulators to it. Then from the G1 or G2 phase cell will start dividing. So it means हमने एक step हासिल कर लिया यहाँ पे. What we did, we take a cell suspension culture in a liquid medium where we have not added growth regulators. So what will happen, all the cells which is present in this liquid medium will get arrested at G1 or G2 phase. So this was the first parameter. When the cells, we realize that there is no further growth of the cells are not growing, we realize that after a certain period of time that the cells are in G1 or G2 phase, what we do, then we add certain growth regulators. Then from this particular stage cell will start dividing. So all the cells will follow the same path and the same cell cycle steps. It means by this way we are able to get the synchronized cultures. Second is by using certain inhibitors. Second technique is by inhibition technique, means we have to inhibit something. What we do, we use certain inhibitors like biochemical inhibitors which will arrest our cells at G1 phase, G1 stage of cell cycle. Second is the inhibition, where we are able to use some biochemical inhibitors which will arrest our cell at G1 phase. Like we can use for DNA, जब DNA synthesis हो रहा है, we can use certain analog of the DNA bases. Like we are having 5-bromouracil, we add 5-bromouracil in this. सो बट इट इज़ जस्ट एज सिंपल, ये ऐड होता है लेकिन उसके बाद इसकी रेप्लिकेशन नहीं हो जाती है, इनकी जीनोम की रेप्लिकेशन नहीं होगी. सो ऑल द सेल्स विच आर प्रेजेंट इन द सेल सस्पेंशन कल्चर विल गेट स्टॉप्ड एट द G1 phase. मीन्स एक जगह स्टॉप हो गई, देन अगेन वी विल remove this inhibitor by changing the medium, then we will place it into another medium, then from the G1 phase the cell will start dividing, and all the cells which are obtained by this technique are synchronous to each other. Okay, these are the basic two techniques which are used for synchronization of the cell suspension culture. Another technique where we have to see the viability test. We have to see the cells which we are going to use for cell suspension culture are viable or not. Because if we will proceed further, we have to make media, we have to keep everything sterile. So this is cost effective to make sure that the cells which are using for further processes are viable. So we are having different techniques by which we will be able to check the viability of the plant cells in the medium. First is simply by using phase contrast microscope. Simply by using phase contrast microscope, as we all have, we all know the principle of phase contrast microscope. These microscope are mostly used for the live cells, where we will be able to differentiate healthy nucleus and the streaming cytoplasm with the dead cell. Like in case of dead cell, there will be no streaming. So by observing the contrast between the two cells, we will be able to compare the culture which we are having in hand, how many cells are dead there and how many are viable live cells. So first test for viability test is by using cells or by observing cells under the phase contrast microscope. वो फेज कंट्रास्ट माइक्रोस्कोप के ज़रिए से हम लोग सेल वायबिलिटी चेक कर सकते हैं. कैसे? जो हमारी हेल्दी सेल्स है उनमें स्ट्रीमिंग साइटोप्लाज्म होगा. वो मतलब जब स्ट्रीमिंग साइटोप्लाज्म है तो उसमें क्या है, मीन्स मूवमेंट होगी ऑर्गेनेलीज की. और जो डेड सेल है उसमें हेल्दी न्यूक्लियस नहीं होगा और न स्ट्रीमिंग होगी. वी कैन डिफ्रेंशिएट टू डिफरेंट सेल्स अंडर द माइक्रोस्कोप. सेकेंड इज यूजिंग इवांस ब्लू स्टेन. Second is by using Evans blue staining. This is homework to you. In next lecture I will ask you principle of Evans blue staining. So this is homework to you. तो what we do in Evans blue staining, ये जो Evans blue है इसमें कैपेसिटी है कि ये सिर्फ डेड सेल्स ही मतलब ये ले लेती है स्टेन ले लेती है. जो लाइव सेल्स है इवान सेल्स नहीं लेती है. जितने हमें दिखेंगे माइक्रोस्कोप के अंडर डॉट्स ब्लू ब्लू ब्लू, उतना मीन्स उतना क्या हमारी सेल डेथ है. जितने कम दिखेंगे सेल डेथ कम है, जितना ज़्यादा ब्लू स्टेन दिखेगा उतने जितने स्टेन ज़्यादा दिखेंगे उतनी सेल डेथ है, और जितना कम दिखेंगे उतनी सेल वायबल है. दिस इज़ होमवर्क टू यू. इन नेक्स्ट लेक्चर आई विल आस्क एनीवन कि इवांस ब्लू जो है वो कैसे क्या वर्क करता है, वाई दिस इवांस ब्लू इज टेकन ओनली बाय द डेड सेल्स. अनदर इज बाय यूजिंग द फ्लोरेसीन डाइएसिटेट. जो फ्लोरेसेंट दिवान इवन सेल्स हैं फ्लोरेसीन डाइएसिटेट. सो व्हाट इज हैपनिंग इन दिस फ्लोरेसीन डाइएसिटेट में क्या होता है? फ्लोरेसीन जो डाइएसिटेट है हमारे साइटोप्लाज्म सेल्स में क्या होता है, एस्टेरेसिस होता है ना, बहुत सारे एंजाइम्स प्रेजेंट होते हैं. जब हम लोग फ्लोरेसीन डाइएसिटेट डालते हैं, एफडीए डालते हैं सेल्स में, जो हमारी सेल्स की एस्टेरेसिस है, दिस ब्रेक्स दिस फ्लोरेसीन डाइएसिटेट इनटू फ्लोरेसीन एंड डाइएसिटेट अलग से. जो फ्लोरेसीन है ये सेल्स ले लेती है क्या, दिस फ्लोरेसीन which is broken by the esterases which is present in the cell. Then this fluorescein is a fluorescent compound. This fluorescent compound is taken by the cells. So by observing this cell suspension culture under UV light, we observe green color cells. We observe green color cells. हम लोग क्या करते हैं, फ्लोरेसीन डाइएस्टेट लेते हैं ये जो फ्लोरेसेंट कंपाउंड ये इस कथ में तरान प्लांट सेल्स मंजन जो ये प्लांट सेल्स है उनमें बहुत सारे एंजाइम्स प्रेजेंट होते हैं one of the enzyme which is utilizing or which breaks this fluorescein diester into the fluorescent compound यह compound जो fluorescein ये cells obtain करा अच्छा ठीक अंडर जब हम लोग इसी सैम्पल को लेंगे यूवी लाइट के अंडर उसको एक्सपोज करेंगे जो ये हमारी लाइव सेल्स है ये अपीयर होती है ग्रीन मीन्स दिस फ्लोरेसेंट डाइस्टेट is the detection test for the live cells as well like Evans blue करना यूज डेड सेल्स के लिए बट इन केस ऑफ लाइव सेल्स ऑब्टेन के लिए हम लोग क्या करते हैं फ्लोरेसेंट डाइस्टेट यूज करते हैं even what we can arrest our cells at mitotic arrest जिसमें बोलते हैं as we know till now that there is a compound called as colchicine which normally arrest our chromosomes in the mitotic phase so उसकी वजह से क्या होता है उसकी वजह से भी एक अरेस्ट मीन सारी सेल जब माइटोटिक स्पिंडल बनाती है मेटाफेज में सारी सेल्स वहाँ तक जब पहुँचती है तब कॉल्टिसिन एक्ट करता है मीन्स अगेन दिस अरेस्ट्स आवर सेल एट अ पर्टिकुलर सेल डिवीजन then again removing the effect of colchicine in the medium then cell will start growing. This was how our synchronous cultures are obtained by using different techniques. Now we have to again now this is synchronous culture मिला now we have to sure जो हमारे पास synchronous culture है वो grow कर रहा है कि नहीं कर रहा है जो हमारे पास synchronous culture whether this culture which we are having are able to grow or not. So for that we are proceeding with the many different tests like first test is the cell viability test. Second we have to check packed cell volume. Third we have to check the cell number because if cells are dividing there will be obviously increase in the cell number. Then we can check this growth of the culture by fresh weight to dry weight or we can check this growth of the culture by medium conductivity. It means जो हम लोग monitor कर सकते हैं cell growth को we can carry this cell growth of plant tissue culture by 5 different techniques. First is the cell viability, packed cell volume, cell number, fresh weight and dry weight, and third is the medium conductivity. I am recording this lecture, don't get panic. अगर ये fast भी जा रही है, you can listen this again and again. As we discussed cell viability, कैसे phase contrast microscope, Evans blue staining. Third is the fluorescein diestate, and the fourth is a mitotic arrest. Means cell viability we have checked by different techniques. Then second is the packed cell volume. So what we do for in second case, but for all these viability test or for the growth for measuring the growth of the culture, we have to be sure that the cell suspension is uniform, like it's not clumps are not there. If there is a formation of clumps, what will happen, the larger sized cells will get at the base, so there will be no uniformity of the cells in the cell suspension culture. Means जिसे cell suspension culture ना check करूँ कत खत growth खतर वो क्या होना चाहिए, यूनिफॉर्म होना चाहिए, उसमें सेल क्लम्प्स नहीं होने चाहिए. अगर ये सेल क्लम्प्स हमारे पास है, व्हाट हैपनिंग, ऑब्वियसली डेंसिटी ज़्यादा है, शेक हो रहा है, वो बेस में रहेंगे, उसकी वजह से हम लोग क्लियर रिजल्ट्स नहीं लेंगे. तो वी हैव टू बी श्योर जो हम लोग सैम्पल ले रहे हैं उसमें क्लम्प्स नहीं है. द सेकेंड टेक्निक इज देन द पैकेट सेल वॉल्यूम. सो व्हाट वी डू, cell obviously we will take a cell suspension culture, we will mix this culture, then we pour 10 to 15 ml of this culture into the centrifuge, big centrifuge tubes. Then with the low rpm or at the low rpm we centrifuge this. So what we will, for example, we have taken the 10 ml in a 50 ml or 10 ml, we have taken a 10 ml test tube in which we have poured a 10 ml liquid cell suspension culture. In this 10 ml cell suspension culture we rotate it with this small मतलब little RPM, not that much, so that cell is not going to break up. तो what is going to happen, अगर हम लोग इस चीज़ को centrifuge कर रहे हैं, the cells which are present in the liquid medium will get settled down and they will form a pellet. So by this way we are able to check how much percentage of cells have occupied at the base. Assume we have taken a 10 ml. In this 10 ml हमने लिया centrifuge tube में, उसको क्या किया, rotate किया to 5 RPM, so that we are sure that all the cells which are present in the liquid medium should get settled down and form the pellet. फिर हम लोग pellet का volume check करेंगे. Then we will check the volume of the pellet. How? For example 10 ml है, उसमें से हमें 1 ml का pellet मिला, तो it means 9:1 ratio आया. 9:1 ratio आया. Again after 24 or 36 hours we will again proceed for the same procedure. तो फिर समझो हमें उसमें से हमने कितना लिया है 10 ml, उसमें हमें 2 ml जो है 2 ml की स्पेस में हमें उसमें सेल्स मिली और 8 ml की स्पेस में हमें लिक्विड मिला. तो इट मीन्स सेल इज इंक्रीजिंग इन देयर साइज बाय ऑब्जर्विंग द इंक्रीज मास in the form of pellet in the centrifuge tubes. Is this clear to everyone till now? Okay, then तो packet cell volume को ना जो आपने already concept you are having for that. Then third is the cell number. Means we have to take a sample, we have to count the number of cells in the sample. So mostly we used to take number of cells per unit volume. Number of cells per unit volume means कितनी cells इस volume में है, then we will cross multiply it by the total number of cells with the total volume. So जो ये हम number of cells है per unit volume, this can be used with the help of using hematocytometer. So जो ये हमारा hematocytometer है, where we are using our blood cell count भी वहीं पे करते थे, या live sperm count भी हम लोग earlier, मतलब this is early, मतलब initial technique. Nowadays we are using cell counters वगैरह वगैरह. जो ये सेल नंबर है हम लोग क्या करते हैं, एक इस जो हमारा hematocytometer है, these are having 4 different corners. So at one corner we will add 0.1 ml of liquid. Then they are having small grid like structures where we are in each grid we have to count the cell number. For example हमें 0.1 microliter में 10 cells मिली, then we will cross multiply it with the whole volume. Then this way we are able to count the total number of cells in a particular medium. But इसके लिए हमें क्या होता है कि we are having certain limitations for this. Sometimes we get confused with the things, or even if cells are not dispersed मतलब uniformly in the cell suspension culture, what we do to make cells मतलब like free from one another, or to break these cell aggregates, we use certain types of acids. So scientists, in case of plant biotechnology, scientists have used 10% HCl, or they can use 10% chromic acid. 10% HCl and 10% chromic acid, which break these cells or aggregates of cells into the single cells. But still these जो ये हमारे acids are, these are toxic to the cells, and fumes will be मतलब अगर HCl खोलते तो fumes आते हैं, तो it's little tricky. But still earlier scientists used to do it for the cell counting method. Then another is a fresh weight and dry weight. Again what we will do for the fresh weight, we take the cells, we मतलब 10 ml for example हमने 1 ml लिया, pre-weighted petri plate लिया हम लोगों ने, उसमें हमने 1 ml डाल दिया, तो चेक किया कितना आया. Then after 2 days 3 days again we take a pre-weighted petri plate, again हमने इसमें 1 ml liquid डाल दिया. Then we see the difference whether cells are growing or not. By मतलब final initial minus finals we give as a total increase in the number of cells in the cell suspension culture. You go fresh weight. वो फ्रेशली सेल्स देवा. इवन इन केस ऑफ ड्राई वेट ऑल्सो व्हाट वी डू, हम लोग प्री वेटेड पेट्री प्लेट लेते हैं. देन वी pour our 1 ml liquid medium onto it. Then we allow this medium to get dried up. We allow this medium to get dried up so that only cells will be weighted. What we do, we place this petri plate in the oven at 60 degrees centigrade for 48 hours. फ्रेश वेट टू सिंपल तुलन चुना. ड्राई वेट में क्या हम लोग करते हैं, प्री वेटेड. दिस मे बी विल नॉट बी प्रेजेंट ऑन योर नोट्स सम, मतलब कुछ पॉइंट्स. प्री वेटेड पेट्री प्लेट लेते हैं, उसमें हम लोग क्या लेते हैं, 1 ml 2 ml लिक्विड लेते हैं. फिर उसके बाद हम लोग इस पेट्री प्लेट को कहाँ रखते हैं, ओवन में एट 60 डिग्री सेंटीग्रेड फॉर 48 आवर्स. फिर अगेन वी वेट द थिंग्स. तो प्री वेटेड पहला वाला जो है, और सेकेंड वाले में वेरिएशन जो आएगी वो सेल नंबर की वेरिएशन आएगी. देन थर्ड इज द कंडक्टिविटी एंड पीएच. जो ये कंडक्टिविटी है इसकी, cells are growing, हाँ, तो we can use conductive meter by which we will be able to see whether the cells which are present in the liquid medium are growing or not. So conductivity कैसे आएगी, when there is a variation in the pH. जब cells होती है ना, जब cells start करती है डिवीजन, जब सेल ग्रो स्टार्ट करती है, उसमें क्या होता है, they produce H positive ions, they produce H positive ions which decreases the pH of the medium, and this decrease in pH is up to 3.5 to 4.5. हम लोगों ने मीडियम लिया, मोस्टली हम लोग लेते हैं 5 to 6 pH. कितना लेते हैं हम लोग मीडियम का pH, 5.5 ऑल्सो लेते हैं, 5 to 5.5 ऑल्सो. मीडिया की बात कर रही हूँ, pH कितना लेते हैं, B5 में, MS मीडिया में, हाउ मच pH we take there? सो जब हम लोग एक स्पेसिफिक pH लेते हैं 5.5, जब सेल की एक्टिविटी स्टार्ट होती है, मीन्स सेल का डिवीजन स्टार्ट होता है, व्हाट इज हैपनिंग, द सेल H positive ions are released in the medium. Then this pH of the medium is decreased 3.5 to 4.5. Then this decrease in the pH is reflected by the conductivity meter. Then again, when the cells has completed the nutrients which are present in the medium, then again there is the rise in the pH from 3.5 or 4.5 to 5 to 5.5. This is how the things work there.